and let's learn about the characteristics of the virus. HIV virus, its capsid is icosahedral, the cubic symmetry. The virus is enveloped, so there is a membrane envelope surrounding the particle. So the virus is roughly spherical, and it belongs to the lentivirus subfamily, retroviruses. Anyone know the meaning of lenti here? Exactly, yeah, slow. So because the virus replicates slower than others, so lenti means slow. Retroviruses contain RNA as genome, and in the viral particle, there is also a DNA polymerase here, this one. So this DNA polymerase is a reverse transcriptase, which can transcribe RNA back to DNA. And in HIV particle here, you would find 2 copies of its RNA. Okay, this is not segmented virus. These 2 copies are identical. Okay, so they are just 2 copies of the RNA genome. The 2 viral strands of RNA found in the core, surrounded by the protein coat. And on the protein coat, there is a capsule protein, P24, okay, which cover the RNA. Okay, just remember the name of P24, because later when we introduce a new medicine to control HIV, it targets on the P24. Are you done? Okay, P24. And on the viral envelope, you would find some important glycoproteins, the spike proteins. The famous one we want to remember is the P120. Okay, P120, this one. This is a ligand which binds to its cellular receptor for attachment. And the other one is a transmembrane protein linked to P120. This is GP41. Okay, GP41. These 2 proteins are important to initiate the infection. Okay, so this is just a general introduction of the structure of the virus particle. This is the genome of HIV virus, HIV type 1. And roughly the genome can be divided into 3 parts. Let's see. The first part, the encoding one, DAG, is short for group-specific antigen. They encode the structural proteins like P17, P24, the capsule protein, and P7, P6. And in the middle, that's the POL, okay, polymerase. Okay, it encodes P66, P51, and these are the subunits of the reverse transcriptase. While on the 3' end here, it encodes a protein called envelope, ENV here, envelope protein. It encodes a big protein called GP160. But GP160 can be further processed by protease to GP120 and GP41. So that's the spike protein. The 3 major parts of the genome. Okay. introduce the two major GP proteins, GP120. Okay, GP120 protrude from the outer envelope, and this is the ligand molecule which attach to the cellular receptor, and in on human cell that's CD4 molecule, and also co-receptors related to HIV infection. I have a quick question for you. What type of cell express CD4 molecule in human? You are working on immunology. CD4 molecule. Which type of cells express CD4? T cell, yeah, CD4 positive T cell. And anything else? No. Macrophages. Yeah, some macrophages also express CD4. So at the early stage of HIV infection, it actually targets on macrophages first. Then it targets CD4 cells. Okay, the other one is GP41, which is a transmembrane protein. Okay, here, the red one. GP41 anchors the GP120 on the envelope, okay? And GP41 can change their structure, the conformational change. And this structural change, conformational change, mediates the membrane fusion of the envelope and the host cell membrane. So that membrane fusion facilitates the penetration of the core into the host cell. So this facilitates the viral penetration. Later, I'll just play a short video on this, how they work together, how they bind to the host cell receptor, and how the GP41 molecule, the protein, changed its conformation to initiate the penetration. Let me show you the video later. These are the two major spike proteins. Then we'll just have a quick recall. We learn about influenza virus. On influenza virus, it also has the spike proteins, the two major ones. What they are? NA-HA, yeah. Okay, and you should know the function of HA and NA. Okay. So we learned that for each virus to start the infection, the first step is attachment. So GP120 binds to its receptor, the CD4 molecule, okay? And then it also binds to the co-receptor. This is just the old style picture. This is not accurate. Let me show you the video. It's more accurate. Okay. It's not GP41 to bind a co-receptor. Okay, this is a viral replication life cycle. Okay. From here, this is attachment. GP120 binds to the receptor, CD4, and co-receptor. Then the GP41 change the structure, the conformational change, to fuse the envelope with the human cell membrane. Then the core, the nucleocapsid, get into the cell, the host cell, and release its RNA. Okay. Well, this is genome, two strands of RNA, and it also carries the reverse transcriptase. So the reverse transcriptase make the DNA strand from the RNA. Okay. Then to make the double strand DNA. After that, the double strand DNA cannot stay in the cytoplasm, right? So it gets into the nucleus. This is a special step. Get into the nucleus, it carries a special enzyme called integrase. The integrase can insert this viral genome DNA into the human DNA. So it just cut the human DNA open and insert the viral DNA into the human genome. Then from this DNA, it can transcribe to make messenger RNA. Then the messenger RNA get into the cytoplasm to guide translation to make the viral proteins. And okay. And after that, the viral can assemble to form new viral particles and release. This is just a rough life cycle. So here, the key point you want to remember the function of the two enzymes, reverse transcriptase, integrase, and after translation, there is also the protease, which can cut the protein into functional units to form mature viral particle. Okay, let me show you the whole the video. In those video, it has updated information. Basically, this is the correct procedure. Okay? That is too simplified. Okay. This picture gives us a direct sense of the size of HIV. HIV, this is just part of a T cell surface. And you can see that the HIV viral particle on the surface is too small, just tiny spots, tiny dots on the human cell surface. Okay. So compared to the host cell, the T cell, HIV virus particles are very small. Okay. We'll introduce a little bit about the pathogenesis and the immunity. The source of transmission, HIV, well, even though now we know HIV virus originate from some primate animal in Africa, but right now it's just a human pathogen, okay? And it does not infect other type of animals. So the source of infection can only be either patients with AIDS, so that's a late stage patient who already developed the syndrome, or those carriers, those virus positive individuals who are already infected by HIV, but they haven't developed any symptoms. So those are the only source of HIV. The transmission routes can be in different ways. Like the most common way right now is sexual contact, either homosexual or heterosexual contact. Even though by the time of discovery in early 1980s, it was mainly homosexual contact, but right now it's different. Like in China, heterosexual contact is the major way. Or it can be transmitted through blood transfusion or blood products. And this was a very serious problem a long time ago, like in 1990s here in China, because at that time, most of the people didn't believe there is HIV infection in China. So they didn't check the blood transfusion. So when you donate blood, they don't have the quality control. So because of that mistake, there was a big problem of HIV spreading due to blood transfusion or blood products. And the hot point is here in Henan province. It causes a lot of death and infection. It can also happen through contaminated needles. So when people use intravenous drug, they use the needle to inject the drug. And you know, those people, they don't have a lot of money. And if they have a little bit of money, they would buy the drug. And they don't even want to buy a new needle every time. So they would share the needle, share the syringe, and that causes a problem. So it causes a lot of HIV infection due to the needle introduction directly. This happens not only in Western countries, but also in developing countries like in China. Later, I'll show you a map of the HIV problem in China. And the hot points in China are those provinces. There might be like drug abuse. Let me show you later. And it can also happen in vertical transmission. The positive HIV positive mom might transfer the virus directly to the newborn baby. So this is a major ways of transmission. Okay, you want to remember this picture, this graph. It's like the course of HIV infection. And let me explain it to you. Okay, the x-axis is the time, but you should keep a look on this. The first part is just weeks. So that's short time after exposure, like this time 0 exposure, till 12 weeks after exposure, after infection. Then the time scale is years, okay? Year 1 to year 11. So this is the time scale. Then you want to check two major parameters. One is this purple line, okay? This is CD4 T cell count in the blood, okay? CD4 T cell count. It's the cell number per microliter of blood. And the green line, okay, this is the HIV RNA copies per ml of plasma. So you can remember that the green line is just the HIV titer, the number of HIV. You don't have to remember the exact number here. Let me explain what happens after a person is infected. After exposure here, let's check. First, let's check the viral titer. So after infection, within a very short time, there is a dramatic increase of virus particles in the blood. So the virus can replicate in human quickly. But after this peak, there is a dramatic drop. So which means that your immune system is working. Your immune system tries its best to control HIV replication. So it drops to low level, okay? This is the HIV load. Then after that, for a long time, this might take 10 years. The HIV virus level can be kept at a low level for a very long time. This time we call it clinical latency. No symptoms for this long period of time. Then after about 9 or 10 years, the viral load would increase again. And at about 10 years, the patient would develop a lot of symptoms. And that is the phase we call AIDS. So AIDS, S means syndrome, a lot of symptoms. So only those late period of patients, we call them AIDS patients. Well, if here, we don't call them AIDS patients. They are just HIV positive. They don't develop any symptoms. So this is the viral load. It takes, without treatment, it takes about 10 years to finally kill a person. Then let's check the CD4 T cell number. After its primary infection here, you can see that the count of CD4 T cell drops dramatically within a short time, okay, several weeks, okay. Then you can sense the struggle of the immune system. The immune system tries to replace the loss of CD4 T cells. So there is a moderate recovery of CD4 T cell number increase, but it cannot get into the original number, original level. Okay, then after that, over a long period of time, about 10 years, the CD4 T cell number would get lower and lower gradually for a long time. When finally the CD4 T cell number is too low, the immune system collapses. It cannot fight anymore. The person would die due to HIV infection. This is the natural course of HIV infection without any treatment. So to help those patients, to help those HIV positive persons, you want to try to, control the viral replication. You want to inhibit the viral replication. So that's how to treat HIV infection. Let me show you later. We actually talked about that before. So this is just this description of the picture we just mentioned. So the latency might last for a long time, long period of time, 10 years. The reason is that the HIV can insert its DNA genome into the human cell and become latent. So they just release viral particles slowly. And that is also one of the major problems, one of the major obstacles that HIV infection is not easy to be treated because it's already inserted into human genome. It's not easy to clean it, to completely clean it. And by the late phase of HIV infection, except for those cancer symptoms, there are also opportunistic infections. If our immune system is working well, we can fight against a lot of infections, such as bacterial infections, especially tuberculosis, fungi infection, or other infections. But if your immune system is compromised, you cannot fight against those infections. So those people would develop a lot of opportunistic infections. The symptoms of the acute HIV infection, so that's short time after exposure, would usually non-specific include the common familiar symptom we know, like fatigue, rash, headache, nausea, and night sweats, which is not specific. So it's not easy for you to decide that you are already infected, or it might be a common cold. And AIDS is characterized by pronounced suppression of the immune system and development of a wide variety of severe opportunistic infections or unusual neoplasm. So that's Kaposi sarcoma, the cancer I mentioned just And with no treatment, the interval between primary infection with HIV and the first appearance of clinical disease is usually long in adults. It takes about 8 to 10 years. Then death might occur after 2 years, after the symptoms develops. So this is the course of HIV infection. This is a Kaposi sarcoma, the gay cancer. They named it in 1980s. Okay, appears on the skin like this. Okay, rare cancer on the skin is actually a blood vessel cancer, not a skin cancer. Okay, blood vessel. It's like this bluish red oval shaped patches on the skin. Okay, it might be like single patches or in clusters. This Kaposi sarcoma. This is the candida infection in HIV positive persons. Okay, this is a human throat with this candida infection on the mucous membrane. Anyone know this pathogen? It's a fungi-like pathogen. It's a eukaryotic fungi, which can cause human infection. It can cause a lot of infection, either the superficial skin infection, mucous membrane infection. It can also cause systemic infection, like infection in the liver and kidney, and also it causes the bloodstream spreading in human. Okay. HIV-infected persons develop both humoral and cell-mediated responses against HIV-related antigens. And most infected individuals make neutralizing antibody against HIV. So their immune system is working directed against the envelope glycoprotein, but those neutralizing antibodies are not fully sufficient because the levels of the neutralizing activity are relatively low, and the virus is always changing. In a person's body for like 10 or 12 years, the virus, the HIV virus in the human body is evolving, is changing all the time. So the antibody previously made cannot target the virus later, okay? So we just mentioned that the human immune system makes a neutralizing antibody, but because of the great sequence variability of the glycoprotein on the surface of viral particle, especially the variability of GP120, the neutralizing antibody cannot protect fully. And the reason behind this great sequence variability of GP120 is due to the property of retrovirus, transcriptase. Okay, the retrotranscriptase is a DNA polymerase, but its proofreading ability is very low. So when it makes DNA from RNA, it's possible that it induces mutations in the sequence. When it's a regular DNA polymerase, when there is a mutation, it will proofread and correct it. But this retrotranscriptase, it cannot correct the mistakes. So that induces a lot of mutation and leading to the great sequence variability. So that is also the reason why till now we don't have vaccines available, because whenever you make a vaccine, the virus is changing so fast. Okay. Here are some methods for laboratory diagnosis of HIV infection. And because of this serious problem, methods have been developed in different ways for diagnosis. No matter you want to detect the presence of antigen or the presence of nucleic acid. Like here are some way you can take, such as virus isolation. Isolate HIV, then co-culture it. Yeah, definitely it's not an easy way, right? You isolate the virus particle, then you need to find the cell line to culture it, and it requires special equipment and it's expensive. But you can also run other ways, like serological diagnosis. Just check the antiviral antibody in the human serum. It's possible. Or that you just measure the viral antigen or the nucleic acid in the bloodstream. It's easier. Like you can detect the presence of the structural protein like P24 or P17 in the bloodstream. And the simple methods have been developed like this, and you can get the results in like several minutes. If you run PCR to detect the presence of nucleic acid, that's also possible. You can detect both the RNA or DNA in the bloodstream. And actually, in our hospital here, the campus hospital, there is a special office, and they provide free service on HIV diagnosis. They have such equipment to help students to check the status of their HIV infection, and it's free. If some students were diagnosed positive, they would report the information to CDC. Then CDC would run more checks, more serious checks to confirm whether a person is positive or not. And once a person is confirmed positive, they would provide a free medicine for the person. So yeah, infectious disease, as we mentioned before, it's not a personal problem. It's a problem of the whole society, of the whole population of humans. So we should do everything to control it. Okay, treatment. Actually, when we talk about viral treatment, we mentioned this before a little bit, such as you can use a nucleoside or nucleotide inhibitor or analog. Okay, nucleoside or nucleotide analog, which can stop the reverse transcription. Or you can use the inhibitor of the enzyme reverse transcriptase, or inhibitors to the viral protease. Okay, because in the viral genome, a lot of proteins are co-transcribed, co-translated, and it relies on the protease activity to cut it into functional proteins. So there are inhibitors designed to those proteases. But the lessons we learned in the last several decades is that either single therapy, the monotherapy, usually results in virus resistance. So results in a rapid emergence of drug resistance due to HIV mutation. So the strategy here is that they use the highly active antiretroviral therapy. So that's combining different medicines with different mechanisms to inhibit HIV. So it's like a cocktail containing several different type of medicines together. So generally for everyday life, we call it cocktail therapy of HIV. It's not a real cocktail. It's just a mixture of medicine so that we can try our best to inhibit the replication of the virus. And yeah, as I mentioned that till now we don't have any commercial vaccine available for HIV. And that's due to the great sequence variability, the high frequency of mutation. Okay, so in 4 decades, HIV problem is still a big problem for the whole human beings. This is a data of 2024, the global data of 2024, showing the population living with HIV. And yeah, a lot of countries data is not available, like in China, in America, in Russia, but the rest of the world, you can see the distribution of HIV. And okay, hotspots is here. Sub-Sahara countries, especially these parts. A lot of people are HIV positive. So it's a human's task to find better ways to control HIV, especially in Africa, this part. And let me show you. But things are getting better than before. And this is the data of the global trends in people acquiring HIV and people dying from HIV-related disease. The blue one, this one is the people newly infected with HIV. From 1990s till now, you can see that the curve is declining. So every year, the number of new infected persons are getting fewer and fewer. Okay, so that's a good thing. And this is the people died due to HIV infection. It's also getting lower. This is a global data of all the people on the earth. But the situation here in China is not that good. Let me show you the data. So this is a map of China showing the hotspots of HIV infection. And you can see that the several provinces with deep color, the Guangxi, Sichuan, Yunnan, Guizhou, and Xinjiang province. These provinces are the hotspots. The major reason of this is that this part, there is the center of drug production. So there might be some hidden routes in those provinces of the drug dealer. So those people there, they might use drugs, injecting the drugs. So there is cross-contamination and spreading of HIV in those areas and this area. Okay, and Henan is here. And after about 30 years, situation in Henan province is getting better. Because in 1990s, there was a serious problem in Henan province. A lot of HIV infection. And that is not due to drug user or sexual contact. That's at that time that Henan here in the Henan province, people were very poor, they regularly donate their blood to companies to get money. So it's actually not donating money, they sell their money. They sell their blood for money, a little bit of money. But those companies collecting the blood are not curious enough. They didn't check the quality of the blood, and it causes cross-contamination. So it causes spread in some cities, some villages in Henan province, causes a lot of HIV infection. But yeah, after that, it's getting better and better. So this is the distribution of AIDS in China. The data in this map is just data for AIDS, but for HIV positive person is maybe big, the number is bigger than that. So this in this picture, let me show you. This is the okay. In each column, the color shows the different transmission route. Okay, and you can see that before 2005, this one, the major transmission is due to the intravenous drug user or the paid blood donor. So that's the major reason. But right now, the latest data is here is 2019. The major reason is this, the blue one is actually the heterosexual problem, okay? And this is the homosexual man, okay? So sexual contact is a major route of transmission right now. But the problem you find in this curve, in this picture, is this curve. It's getting higher and higher. This is a newly reported HIV/AIDS cases. It's getting higher and higher in China. So the situation is not good. It's getting worse and in China. So hopefully we can do something to block HIV spreading in this country. There is some data saying that the positive rate in China is about 1 out of 1,000, and this number is very big. 1 out of 1,000 in China, the whole population in China. Okay. Okay. With the rest of the time, I'd like to introduce Dr. Gao Yaojie to you. And Dr. Gao actually graduated from our medical school, one of our famous alumni. She was born in 1927 and passed away a couple of years ago. She worked as a doctor in Zhengzhou City. Okay, in Zhengzhou City, she worked as a doctor in a hospital. After her retirement, you know, in China, people retire when they are 60 years old. After her retirement, in that hospital, the doctors found some weird disease, and they couldn't recognize the type of disease. That was in 1990s, and Dr. Gao then noticed that the disease is actually AIDS, HIV infection. And after that, you know, she retired. So she tried to began to investigate HIV infection in Henan province by herself, without any support, without any finding. She just worked on that by herself. And she later found out the real problem is due to the paid blood donor in some villages in Henan province. And you know, this kind of investigation touches others' cakes. So the companies didn't like that. The government didn't like that. So she was, for a long time, a not welcome person. And some time ago, she went to America, and she lived in New York City for a long time. And she passed away in New York in 2023. A very great person, and who did important and interesting work for people here, but she was forced away from China after that. Very brave person, Dr. Gao. Okay, good news. The recent advancement of AIDS therapy or HIV therapy. This is a new medicine. The name of it is lenacapavir. Okay, this is a structure of the medicine. This medicine actually targets on T24. You mentioned that the capsid protein T24 is a capsid inhibitor. Okay, here it inhibits the nuclear transport, inhibit the viral assembly, and also inhibit the formation of the capsid. Target binds to T24, and it was approved as a medicine for HIV treatment. Okay, if a person, the virus in a person's body is resistant to other drugs, they can try to use this medicine. But that's not the good news. The great news is that in last year, they did some clinical trial in Africa. They used this molecule, this medicine, as a pre-exposure prevention. Okay, pre-exposure HIV prevention, short for PrEP. So they give it a short injection in women in Africa. They only inject the medicine twice every year, so twice yearly into the person. And it turns out that these 2 injections can prevent almost 100% of HIV infection in those clinical trials. So this is very effective, even though it's not a real vaccine, but it can block HIV infection. So this work, okay, 99.9 efficacy. So this work was named as the 2024 breakthrough of the year by the Journal of Science. So hopefully with this medicine, we can finally block the spreading of HIV infection. And there are some other work, and I shared in my WeChat that some group did immunotherapy of HIV positive persons. They use neutralizing antibody and also stimulate CD8 positive T cell. And after that, they can inhibit replication of HIV for several months without any medicine. They can activate the immune system to control HIV, which is a new work recently published. Okay, this is a new medicine. Hopefully we can use it in future. Okay, I just mentioned that the life cycle of HIV, how the virus attaches to the host cell, how it replicates, and let me show this video.
So this is HIV. It's a typical retrovirus, meaning that it has an outer envelope And in the center, it has 2 copies of RNA, as well as an enzyme here in blue that's reverse transcriptase, which will ultimately turn that RNA into DNA. The virus itself, with this outer envelope protein, actually directly infects T helper cells. The way that it does this is that as it comes up to the cell surface, it uses receptors that are on T helper cells and exclusive to T helper cells, which are CD4 molecule, which really defines T helper cells. It's a surface receptor that binds to the envelope protein. That causes a conformational change and allows a second receptor to grab hold of the envelope. This is the chemokine co-receptor. It's also called CCR5, and we'll talk about that more. What happens now is that the stalk of the envelope protein pierces through from the virus into the host cell and starts to draw the cell membrane and the viral membrane together. And what ultimately happens is fusion of those 2 membranes, and the viral genetic material is injected essentially into the cell, and the envelope protein is left at the cell surface. The virus has a matrix and a capsid protein, shown here in green and red, that essentially are digested when it enters into the cell. That releases viral enzymes in the viral RNA. And here we have reverse transcriptase, which takes the viral RNA and, using host nucleotides, converts that viral RNA into a single strand of DNA. While it does that, it makes some random errors, which is characteristic of reverse transcriptase. It has very poor proofreading activity. That single-stranded DNA now is again reverse transcribed into a double-stranded DNA. At that point, another enzyme that has come in with the virus in the beginning, called integrase, essentially grabs hold of that double-stranded DNA and carries it through a nuclear pore into the nucleus of the cell. Within the nucleus of the cell, it finds the host chromosome, and the integrase enzyme makes a nick in the host DNA and allows for HIV to insert itself into the host chromosome. And that right there is what establishes lifelong infection. Now, RNA polymerase comes along and makes messenger RNA. Those messenger RNAs encode for different viral proteins. They end up associating with ribosomes at the surface of the rough endoplasmic reticulum. And here's a piece of mRNA that's making envelope protein, which is directly produced into the endoplasmic reticulum. And it's shuttled then through the endoplasmic reticulum and taken to the cell surface, where at the cell surface it becomes embedded in the cellular membrane, and at this point coalescing with other envelope proteins that have been produced. You have this cluster of envelope proteins now on the surface of this infected cell. At the same time, there are other messenger RNAs that are being produced that allow for translation of other viral proteins. So here are additional viral proteins being made, which are going to be used to make up the key components that the virus ultimately is going to need. These are transported again to the cell surface, to the area where these envelope proteins are, and a strand of RNA as well as some of the enzymes are part of that complex. This then buds off at the cell surface at this point, but it's still not a mature virion because the polyprotein chain needs to still be digested into its component parts. That's done by an enzyme called protease. Protease breaks up those polyprotein chains and ultimately allows for them to coalesce and form the mature structures that make up the final virion. And now you have a mature infectious virion that can go on now to infect other cells. Once that happens, now the cell can produce tons of viruses, and this is really what then keeps the whole process going.
Okay, so we just introduced the essential information on HIV. And in our PPT and also in this video, it mentioned that there are actually two co-receptors of HIV. Anyone remember that? The major receptor of HIV is CD4, right? CD4 is the receptor, but it also requires another co-receptor. The co-receptor can be CCR5 or CXCR4. There is something interesting for the co-receptor CCR5. It's a chemokine receptor, CCR5. CCR5, actually in human population, there are different alleles. In some people, their CCR5 is actually called CCR5 delta 32, which means that there is a deletion of 32 nucleotides. So it's a mutation gene. In this mutation gene, they couldn't make real CCR5 protein, delta 32. So those people couldn't make mature CCR5 molecule. And it might be not good for other infections, but for HIV, this person with a mutation of CCR5, they are naturally resistant to HIV infection. So this happens with a higher ratio in Europe in those people. And they discovered the presence of this mutation gene of CCR5. There was an interesting story about a patient, they call it a Berlin patient. The person actually was from America, who was AIDS patient. Actually, he was infected by HIV. And unfortunately for him, later he developed blood cancer. And he went to Berlin, Europe, for stem cell transplantation. And it was interesting that after stem cell transplantation, doctors found that he was free from HIV. So after stem cell transplantation, the HIV virus was gone. And then they figured out that the donor cell of the stem cell is delta 32, the CCR5 delta 32, which is naturally resistant to HIV infection. So that's the story of discovery of CCR5 and delta 32 mutation. Several years ago in China, there was a crazy scientist called He Jiankui, and he definitely knew this story, and he saw that, well, can we create a person with mutation of CCR5? So he used the CRISPR gene editing method to edit the embryo of human, and he finally made 2 baby girls with deletion in the gene, CCR5 gene. And after that, he was sentenced to prison for 3 years, and he is out of prison now. But definitely, it's not okay to do this, right? You cannot edit the human genome. You cannot edit the human embryo gene, and it's not allowed. And when we learn medicine, we should have some course related to ethics, right? Ethics? No? Yeah, you should have some course, some training on ethics. And when you are a doctor, when you are like a dentist or a pharmacist, you should know the regulation, what you can do, what you cannot, which is not allowed, right? Ethics. Okay, so basically that's all for today and for this year. Okay, if you have any question, just come and talk.